Welcome to this lecture on the fundamentals of genetics. Today, we are diving into a truly fascinating topic: spontaneous and induced mutations. Imagine your DNA as a massive 3 billion word encyclopedia that holds the instructions for your entire body. What happens when there is a typo in that encyclopedia? How do these typos occur naturally? And how does our environment force these errors to happen? By the end of this video, you will understand the fundamental mechanisms behind genetic variation, how we calculate mutation rates, and why these microscopic changes are the driving force behind both devastating diseases and the magnificent evolution of life on Earth. Let us get started. To begin our journey, let us understand what spontaneous mutations are. Imagine you are tasked with copying a massive 3 billion word encyclopedia entirely by hand. No matter how careful you are, you will eventually misspell a word, skip a line, or accidentally swap 2 letters. This is exactly what happens inside our cells every time they divide and copy their DNA. Spontaneous mutations are naturally occurring changes in the DNA sequence that happen without any exposure to external mutagenic agents. The cellular machinery that copies DNA is incredibly accurate, but it is not absolutely perfect. Sometimes, without any outside interference, a random mistake slips through the proofreading process. These unprompted typos form the baseline of genetic change. Now that we know these natural typos exist, we must ask, what exactly causes them? Spontaneous mutations primarily arise from endogenous biological processes. For instance, the enzyme DNA polymerase can simply make an error during DNA replication. But mutations can also occur due to spontaneous chemical changes in the DNA bases themselves, even when the cell is not dividing. A common chemical change is depurination, where a purine base is randomly lost. Another is deamination, where an amino group is removed from a base like cytosine, turning it into uracil. There is also a fascinating mechanism called a tautomeric shift, where a DNA base temporarily assumes a rare structural form, leading to incorrect base pairing. These events are driven by basic thermodynamics. Understanding these mechanisms leads us to an important realization about their impact. You might initially think that mutations are always bad, but spontaneous mutations are actually the ultimate source of all genetic diversity on Earth. Because they are driven by normal cellular metabolism, they occur at a constant, albeit very low, background rate in all living organisms. Without these random changes, every organism would be a perfect clone of its ancestors, and evolution would completely halt. They provide the raw material upon which natural selection acts, allowing species to adapt to changing environments over millions of years. However, in the short term, they can also be the underlying cause of spontaneous genetic diseases and some forms of cancer. To quantify how often these natural errors occur, we use a specific mathematical representation. This formula helps us understand the probability of a gene mutating under normal conditions. The symbol mu represents the mutation rate, which is the probability of a mutation occurring per cell division or per generation. The variable m stands for the total number of new mutations observed in the population. Finally, n represents the total number of individuals, cells, or alleles examined in the study. In plain English, this formula tells us the fraction of the population that acquired a new mutation. If the number of mutations, m, increases while the population n stays the same, the mutation rate mu goes up. Conversely, a massive population with very few mutations means the rate is extremely low. Before we move on, let us see this formula in action with a concrete example. Suppose a geneticist is studying a culture of E. coli bacteria to determine how often a specific gene mutates spontaneously to confer antibiotic resistance. We are given that out of 50 million bacterial cells, exactly 15 cells have developed this spontaneous mutation. We need to find the mutation rate. We substitute 15 for m, our numerator, and 50 million for n, our denominator. Dividing these numbers yields 3 times 10 to the power of -7. This means that for every 10 million bacterial cells, we expect to see about 3 spontaneous mutations for this specific trait. This incredibly low number highlights just how efficient the cell's DNA proofreading mechanisms are under normal conditions. Now that we have explored natural mutations, let us look at what happens when the environment interferes. Imagine you are copying that same encyclopedia, but this time someone is flashing bright strobe lights in your eyes and bumping your elbow while you write. You are going to make a lot more mistakes than you did sitting in a quiet room. In genetics, this is the intuitive concept of induced mutations. When an organism is exposed to harsh environmental factors, its DNA gets battered and bruised. The cellular machinery tries to copy the damaged DNA, but the environmental interference causes it to make errors at a much higher rate than normal. These changes are directly caused by exposure to external agents, which we call mutagens. Building on the concept of mutagens, let us categorize the specific agents that cause this damage. Mutagens can be physical or chemical. Physical mutagens include ultraviolet light, which causes adjacent thymine bases in the DNA to fuse together inappropriately. Another physical mutagen is ionizing radiation, like X-rays, which are so energetic they cause severe double-strand breaks in the DNA backbone. On the other hand, chemical mutagens include base analogs that mimic normal DNA bases but pair incorrectly during replication. There are also alkylating agents that add bulky chemical groups to the DNA structure. When the cell attempts to replicate or repair this chemically or physically damaged DNA, the normal base pairing rules are disrupted, leading to insertions, deletions, or substitutions. To truly grasp how physical mutagens alter DNA, let us examine this diagram of a thymine dimer. Here, you are looking at a standard DNA double helix. Notice the sun icon at the top, representing incoming ultraviolet radiation. As the UV rays hit the DNA strand, they impart energy that causes 2 adjacent thymine bases, labeled with the letter T, to form an abnormal covalent bond with each other, rather than pairing with the adenine bases across the helix. Look closely at the red highlighted region. You can see how this unnatural bond creates a physical bulge or distortion in the DNA backbone. When the cell's replication machinery encounters this bulky distortion, it often stalls or inserts the wrong bases, directly inducing a genetic mutation. Having seen how mutagens damage DNA, we must ask, how do we measure their severity? To understand how dangerous a mutagen is, scientists calculate the fold increase in the mutation rate. This mathematical comparison tells us how much the environment has amplified the natural error rate. In this equation, F represents the fold increase, which tells us how many times more likely a mutation is to occur in the presence of the mutagen. The symbol mu subscript I is the mutation rate measured after the organism has been exposed to the mutagen. The symbol mu subscript S is the natural background mutation rate we calculated earlier. If the induced rate is much larger than the spontaneous rate, the fold increase F will be very high, indicating a highly dangerous and potent mutagen. Let us apply this fold increase formula to a real-world laboratory scenario. A researcher is testing a newly synthesized chemical dye to see if it is mutagenic and potentially dangerous to humans. We are given the known spontaneous mutation rate for a yeast population, which is 2 times 10 to the negative 6. After exposing the yeast to the chemical dye, the new induced mutation rate is measured at 8 times 10 to the negative 4. We need to calculate the fold increase. We plug the elevated induced rate into the numerator and the natural spontaneous rate into the denominator. Dividing these scientific notations gives us exactly 400. This means the chemical dye causes genetic mutations to occur 400 times more frequently than they would naturally, strongly indicating it is a severe health hazard. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that spontaneous mutations are the rare, unavoidable typos made by the cell's own machinery. While they can cause disease, they are fundamentally the slow and steady fuel for all evolution. Second, we learned how to calculate the mutation rate to measure the probability of these natural errors. Third, we explored induced mutations, which are the rapid, frequent errors caused by external mutagens like UV radiation and harsh chemicals. Finally, we used the fold increase formula to quantify exactly how dangerous a mutagen is by comparing the damaged state to the natural baseline. You now have a solid understanding of the fundamental forces that alter our genetic code. Excellent work.